This is the third video in a series of videos for genes and patterns of inheritance. In the last video, we looked at Gregor Mendel and his first law of inheritance, which stated that factors would segregate out. In other words, each characteristic is controlled by a pair of factors, and when gametes are formed, they separate out. Only one of each factor is found in the gamete, and they do not blend together. In this video, we're going to be looking at some examples of when there's exceptions to this rule. So the first example here is codominance. Okay, and codominance is if two alleles are both expressed in the phenotype, they're described as being codominant. So in other words, there is a little bit of a blend here. Both factors are having an effect and one allele is not being masked. So in all the examples we've looked at so far, the genes have interacted in such a way that one is completely dominant to the other, and the other being the recessive allele. When there's complete dominance over one allele over the other, the heterozygous phenotype is indistinguishable. In other words, it looks exactly the same as the homozygous dominant plant. And that's what we've seen so far, and that's what you would have seen in fifth year biology. When codominance occurs, there is a difference between the heterozygous phenotype and the homozygous dominant phenotype. Okay, so in some cases, the allele of a given gene interact in such a way that the complete dominance is lacking. Both alleles are expressed, and there's going to be a difference. This genetic situation is known as codominance, and in certain cases, incomplete dominance. So in flowers, this is quite common actually. So in snapdragons, we've already seen in the first video that there's 3 different phenotypes. They can be red, they can be white, and they can be pink. Individual plants that are homozygous will be either red or white. Okay, and note here that there's not homozygous dominant or homozygous recessive. Both of these alleles are dominant. So whenever there's one of each, they will act in a codominant way. So the heterozygous plants are not red or white, they're in fact pink. So there's kind of a mixture of the two. Both phenotypes are expressed. When complete dominance is lacking, upper and lowercase letters are not used to note the alleles involved. In these cases, the gene is represented by a capital letter and the alleles as superscripts. So you notice here we've got a capital letter C, we've got this R here, and this W here, and their superscript. R standing for red and W standing for white. Therefore, the red flower will have 2 R's, the white flower 2 W's, and the pink flower 1 of each. So here we have a total of 3 phenotypes. So this doesn't just happen in flowers. Codominance can also occur in chickens. So in domestic fowl, a gene with 2 codominant alleles is responsible for the determination of feather colour. In pure breeding, black feathers colour is due, again, we've got this capital C and the superscript B, and the B obviously standing for black. The white strain will have the W for white genotype. The first generation, resulting in a cross between a black and a white feathered fowl, is made up entirely of slate blue offspring. So here we have our pure black here, and it has got 2 versions of that gene, which are black. The white CWCW, and the first generation results in one of each of these being inherited, and rather than it being black or white, is actually a mixture, which is blue. So if we look at the second generation, the blue slate fowl will produce gametes with either the black or the white allele. So if two of the black genes come together, we result in a black fowl. If one of each comes together, in these two cases here, we end up with a blue fowl. And if the two whites come together, we end up with a white fowl. Again, we get this 1 to 2 to 1 relationship. This ratio has resulted again. This is an expected phenotype for this scenario. A second exception to the rule is multiple alleles. So the genetic system considered so far have been limited to genes with pairs of alleles controlling specific characteristics. Genes are changed to alternative forms by the process of mutation, and it is possible for a given gene to have more than 2 alleles within a population. So whenever there are more than 2 alleles for a particular gene within a population, we have a number of multiple allelic series. An example of this is to do with multiple alleles. So although there may be more than 2 alleles of a gene present within a population, the maximum number possessed by any individual is going to be 2. The reason being, we have 2 versions of each. As we inherit, our chromosomes come in homologous pairs. A classic example of this is to do with blood groups. Okay, so the gene controlling the ABO blood system has 3 alleles within each allele coding for the production of a different antigen on the membranes of red blood cells. So as a result, we can end up with the A blood group, AB blood group, B blood group, or the O blood group. The 3 alleles containing the expression of 4 blood groups are, they have a gene for A, a gene for B, or a gene for neither. That's known as O. So A will produce the antigens on A. B codes for the production of antigens of a different type. And O does not code for any antigens on a blood cell. A and B are both codominant and are dominant over O. As a result, if an individual contains 2 A's or A and O, they will have antigens A on their red blood cell and they will be blood group A. If they can have 2 B genotypes or BO, they will be blood group B. If they contain one of each, they will be blood group AB and have both antigens present on their red blood cells. And if they have both recessive O alleles, they will be blood group O. This results in this case, 4 different blood groups in humans: A, B, AB, and O. Those in A, there's 2 versions. Likewise for B, there's 2 versions. For blood group O, they have to have both recessive alleles. And for AB, they need to have one of each. So we're going to consider the following problem here. So we have a mother who is group B and heterozygous, and a father who is group A and heterozygous. So given this situation, we need to consider the possible outcomes of the children being born to this couple. So as they are both heterozygous, That means they have one of each type of gene. The mother, who is group B, has the recessive O allele and the dominant B allele. The father has the dominant A allele and the recessive O allele. So try this cross and then continue the video on. See if you've done it correctly. So if you've paused the video and tried this, now you can watch on and check your work. So one possibility is that the A and the B, both dominant alleles, come together, resulting in blood group AB. There is a chance that only the dominant B is inherited from the father and the recessive O is inherited from the mother, resulting in blood group B. Likewise, blood group A is possible, A coming from the father and O coming from the mother. And finally, group O, where the child has received blood group O from both parents. So for each individual child, the probability of being blood group AB, B, A, and O is 1 in 4, or 25%. And finally, the last exception to the rule is lethal alleles. So there are many examples within living organisms where a single gene may affect a number of different characteristics, including mortality. Normal wild type of mice have a grey coloured fur, but occasionally mice with yellow fur appear in the population. A French geneticist performed crosses with these yellow and grey mice and found that the allele covering yellow fur is dominant and that yellow mice were always heterozygous. When the heterozygous mice, big Y, small y, were mated together with there was a 2-to-1 ratio found, which is not the expected 3-to-1 ratio. It was eventually found that pure breeding homologous yellows were never found in the population. So there was no such thing as a mouse with big Y, big Y. And this is due to the fact that this allele in the homozygous condition is lethal. They do not exist, they cannot survive. This was confirmed when dead fetuses were found in the uteri of yellow females. So the cross is as follows. So whenever the heterozygous version of each were crossed, the big Y did result. However, due to the fact that this is a lethal combination, this mouse cannot survive. So it will die before it's even born. We have two yellow mice, which are both heterozygous, and then we also have this grey mice. As a result, this one is not living. We're left with 3 in total, which is leaving us with a 2-to-1 ratio. The gene for yellow colour is lethal in the homozygous form. A ratio of 2-to-1 is found, and the Y allele is dominant in its effects, meaning the heterozygous mice are yellow. So that's it for the third part of this topic. Quite a lot in this video to look at. So just to summarise, there was 3 exceptions to Mendel's first law of inheritance. The first law, or the first exception, is codominance. Whenever both phenotypes are expressed in the heterozygous form. The second is multiple alleles. We give the example of blood types there. And thirdly, the last one we looked at was lethal allele combinations, and these all break the first of Mendel's laws, and they are exceptions to that rule. So moving on, next day to be looking at dihybrid inheritance. And you'll find out that the next day. Okay, so good luck, and hopefully that is helpful.